The dire wolf, a legendary predator of the last ice age, is no longer a creature confined to the fossil record. Scientists have brought back the massive canine, marking the first successful de-extinction event in history. This groundbreaking achievement raises profound questions about our relationship with the natural world and the power of science to reshape it. The dire wolf, known scientifically as, roamed North America until around 13,000 years ago. Larger and more powerfully built than gray wolves, dire wolves were apex predators, hunting large prey like horses and bison. Their extinction, along with many other large mammals at the end of the Pleistocene epoch, left a void in the ecosystem. Today, 3 dire wolf pups, born through the efforts of colossal biosciences, represent a bold step into the uncharted territory of de-extinction. These pups are not perfect replicas of their ancient ancestors, but rather the result of a complex genetic process involving ancient DNA and cutting-edge gene editing. This achievement opens up a new chapter in conservation, offering hope for the revival of lost species and the restoration of damaged ecosystems. The journey to bring back the dire wolf began with fragments of ancient DNA. Scientists extracted genetic material from a 13,000-year-old dire wolf tooth and a 72,000-year-old skull. This genetic blueprint provided the foundation for recreating the extinct species. However, reconstructing a complete genome from ancient DNA is a monumental challenge. To overcome this, scientists turned to the dire wolf's closest living relative, the gray wolf. By comparing the genetic makeup of both species, they identified key genes responsible for the dire wolf's unique traits, such as its size and predatory adaptations. Using CRISPR technology, a revolutionary gene editing tool, scientists made precise modifications to the genome of gray wolf cells. A total of 20 specific edits across 14 genes were introduced, nudging the gray wolf's genetic code closer to that of its extinct relative. These modified cells were then used to create embryos, which were implanted into surrogate gray wolf mothers. The result? 3 healthy dire wolf pups, each carrying the genetic legacy of their ancient ancestors. The dire wolf holds deep cultural and spiritual significance for indigenous people, particularly the Mandan, Hidatsa, and Arikara (MHA) Nation of North Dakota. For the MHA Nation, the dire wolf is a revered creature, often depicted in their traditional stories and artwork. Recognizing this profound connection, Colossal Biosciences consulted closely with the MHA Nation throughout the de-extinction process. This collaboration ensured that the project respected the cultural values and beliefs surrounding the dire wolf. The MHA Nation's involvement highlights the importance of incorporating indigenous knowledge and perspectives in scientific endeavors, particularly those involving culturally significant species. The resurrection of the dire wolf has ignited a global debate about the ethics of de-extinction. Critics raise concerns about the welfare of the animals brought back, their potential impact on existing ecosystems, and the possibility of unintended consequences. Others argue that the benefits of reviving lost species, such as restoring ecological balance and preserving biodiversity, outweigh the risks. The successful de-extinction of the dire wolf marks a pivotal moment in the history of conservation. It demonstrates the potential of cutting-edge technologies like gene editing and ancient DNA analysis to not only study the past, but also to reshape the future of life on Earth. The techniques developed for the dire wolf project have far-reaching implications for the conservation of critically endangered species. Scientists believe that similar approaches could be used to revive extinct animals like the passenger pigeon and the woolly mammoth. Furthermore, this technology could help bolster the genetic diversity of dwindling populations, making them more resilient to diseases and environmental changes. For example, scientists are exploring the possibility of using gene editing to save the American chestnut, a tree nearly wiped out by a fungal blight in the early 20th century. The successful de-extinction of the dire wolf serves as a powerful reminder of the potential of science to correct past ecological wrongs and forge a more sustainable future. However, it also underscores the importance of proceeding with caution, carefully considering the ethical implications and potential consequences of our actions. As we venture further into the realm of de-extinction, we must do so with humility, responsibility, and a deep respect for the intricate